Hello, everyone. My name is Hassoon Sarwar,
I'm Emily Lore.
I am Joshua Bain.
and this is our project on SNP identification and analysis.
So the main projects and goal and purpose of this assignment was to analyze and interpret ancestry testing data to make predictions about a disease and a trait risk, research and find which nucleotide at the SNP involves disease risk, research how genetic changes lead to a cellular effect that can cause disease, and research which genotypes and ethnic backgrounds are at a higher risk for certain diseases. So what is an SNP? An SNP is a single nucleotide polymorphism, and it is a DNA sequence variation that occurs at single nucleotide and is found in at least 1% of the population. The SNP we had was RS231775, and that is associated with the CTLA4 gene, and that is responsible for encoding for a T cell protein that regulates immune response. Not having this is an increased risk of autoimmune thyroiditis and variants such as Hashimoto's thyroiditis and Graves' disease. So thyroid hormone controls metabolism, growth, and other bodily function. When you have autoimmune thyroiditis, it leads to the immune system malfunction, where antibodies attack normal thyroid tissue, causing inflammation. Hashimoto's thyroiditis is a decreased thyroid hormone production and is known as hypothyroidism. And then Graves' disease is an increased thyroid hormone production. It is known as hyperthyroidism.
So focusing a little bit more on the molecular genetics of this specific SNP, this SNP involves A to G transition or adenine to a guanine transition at position 49, a polymorphism in exon 1 of the CTLA4 gene, which results in an amino acid change from a threonine to alanine at position 17. And as you can see in this figure right here, this specific SNP RS231775 is located on exon 1, which is the leader peptide in this CTLA4 gene. So CTLA4 is the cytotoxic T lymphocyte associated protein, and it is located on the human chromosome 2 at position Q33, and it is a very common genetic marker for autoimmune diseases. The specific molecular mechanism that takes place with this gene and protein is that this gene codes for a protein that sits on the surface of a T cell, which is an immune cell. When CTLA4 binds to B7, which is another protein, it leads to an inactive T cells and allows for tumor growth and progression. But if CTLA4 is blocked by an antibody, that leads to an active T cell and tumor cell death. In the case of autoimmune thyroiditis, there are antibodies that are blocking CTLA from attaching to the attaching, which leads to inactive T cells and which allows the immune cells to kill normal, healthy thyroid tissues. The methods that we employed in this project was that we first wanted to find an SNP that was associated with pancreatic cancer. We found that this specific gene, CTLA4, has an SNP on its exon 1, and we first searched SNpedia for this SNP, and we learned that this SNP is also associated with autoimmune disorders, specifically ones that are thyroid related. We then found the RSID of this SNP from the ancestry testing data of an anonymous donor, and we found that this anonymous donor had this specific mutation on chromosome 2 at this specific position, and their genotype was a GG, or a guanine paired with a guanine.
Going over the results that we have, as we saw, the SNP that was affected is located on chromosome 2, and the genotypes related to this disease consist of the genotypes AA, AG, and GG. If we look at the table, we see that the genotype AA has no increased risk of autoimmune thyroid disease, while AG and GG both have risk. While AG has a 1.5 times risk of autoimmune thyroiditis, GG has a 2.3 times risk of Hashimoto's thyroiditis and a 1.47 times risk of Graves' disease. We can see that the genotype GG is more risky compared to the other genotypes, and our donor is more likely to have this specific genotype. In this graph here, it shows that the AA genotype is most commonly found in Tuscany, Italia, with 52.5% of the population having this specific genotype. We also see that AG is most common amongst the African ancestry within the southwest of the USA. The genotype GG is most common within the Han Chinese population in Beijing and Chinese descendants living in Denver. They both had about a 47.1% population with this genotype. Using this information and the data sets that were given to us, we can see that the donor has a GG genotype, which is most commonly found within the Chinese ancestry. The donor also has 2 X chromosomes, showing that it is a female. Females are also more likely to develop this autoimmune and thyroid-related diseases, and based on our analysis and referencing our data sets, we can therefore predict that this donor is probably female and of Chinese ancestry.
So after completing this project, there were a few new skills that we gained, and one of them was that we learned to analyze genetic and ancestry data sets in these large data sets that contain genetic information.
We also learned how to understand the relationship between genetic alterations and health risks.
We also learned how to explore molecular mechanisms underlying certain diseases, as well as the clinical relevance of acquiring these skills.
So as biology students some of us who hope to pursue a career in medicine, it's very important to understand what genetic mechanisms and what specific genetic conditions and ethnic backgrounds can influence or increase someone's risk of developing certain diseases. Thank you so much for listening to our presentation. These are our references, and thank you.